Got my support staff in the back. All righty, good morning, everybody. My name is Dallin Stokes. I am one of Dr. Lane's grad students, and I'm sorry you're stuck with me, but Dr. Lane's out of town, so bear with me. We're gonna try and have fun with this. So today, Dr. Lane said I could talk about whatever I wanted. So we're gonna talk about the coolest aspect of viruses, and that's viral vectors. And of course, if you're talking about something called a vector, you can't help but bring up vector. I mean, Victor is my nerd name. Now I am Vector, committing crimes with both direction and magnitude. Oh yeah! So there you go. We're talking about vectors. What is a vector besides a villain in an orange suit? Anybody know? In biology, not physics. Chirp, 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 chirp. Okay. A vector is the method of delivery for a gene into a target organism during genetic engineering. So it depends on what aspect of biology you're working with. That alters the definition of a vector. So if you're talking about like ecology, they would say like a mosquito is a vector for malaria because it delivers malaria to the target organism. In case of genetic engineering, it's whatever you use to deliver a gene from outside of a cell that you're trying to change to the inside of a cell that you're trying to change. And there are lots of different kinds of vectors that we use. Plasmids, if you guys have talked about those before, they're circular loops of DNA that we can transform into bacteria that we can use to carry genes. Those ones we most commonly use on bacteria, but today we're going to talk about viral vectors. So if a vector is a method of delivery for a gene into a target organism, what do you suspect a viral vector is? Yes, on the front row.
Delivery of a gene into a, well, I guess for a virus into a host.
Very good. We're actually going to use a virus to deliver the gene. The virus is the mailman because we're trying to get a gene inside of a cell in order to change it for therapeutic purposes or whatnot. The tricky part is getting it into the cell. And so we're going to use the virus as the delivery mechanism. He's going to be the vector. Now, why would you use a virus to do this? Let's work through some basic theory, and I like to draw, so bear with me here. This is going to be fun. That's a cell. Okay, there's your nucleus. You got your DNA in here. Oh, please no. It's going to do this today, is it? Okay. If everything doesn't explode by the end, we'll call it a success. Okay, so here's my cell. And viruses, as a little bit of a refresher course, here's my little virus. Isn't it lovely? We've got like a little, boop, it's got its little spike proteins poking out on the outside. A virus, if you boil it down to its deepest essence, it's essentially a protein box with DNA or RNA tucked on the inside. That's essentially what a virus is. And viruses work because they can't make their own babies. They have to get inside a host cell in order to make new babies. So they work on a little Trojan horse mechanism. The cell has receptors on the outside that are used for cell signaling and whatnot. They receive a signal and then react to it. Viruses work by building spike proteins on the outside that look a lot like these signal proteins, so that when they bind to the receptor, the cell brings it on the inside, and then the virus cracks open and it's like, bah, make babies, right? That's how viruses work. Well, what we've got is a naturally occurring system for delivering genetic material to the insides of cells. So what we can do is hijack this. What if we crack this virus open, rip out its evil DNA, and instead replace it with our gene of interest? Let's say this is a cell for a person that's got cystic fibrosis, right? So they've got a mutated gene in their cell that's causing this problem. Well, what if the way we could fix that is to deliver a working version of the gene to their cells so they can build a functional protein that would completely resolve their symptoms? So I can rip out the bad virus DNA, slap in the good gene on the inside of this thing, and close the box back up. Now, it still looks like a virus. You got little spikes on the outside and everything. The virus doesn't know the difference. It's just a protein box. And so you put this into the individual, it still goes in there and it cracks open. It's like, ha, make babies. And it's like, psych, you just dropped off the gene for us. Ta-da. So that's the idea behind using viral vectors as a mechanism, is we've got a naturally occurring system for delivering DNA. We're just swapping out what DNA it's actually delivering. Ha, the spacebar worked. Glorious. Okay, so every viral vector ideally has 4 major characteristics. Safety, of course, is highly important. Most viruses are pathogenic. They hurt people, and doctors don't usually like to hurt people. It's kind of in their Hippocratic Oath or whatever, all y'all pre-doctor people. I probably said that wrong, didn't I? Good thing I'm not a doctor. So safety, I mean, we're working with these potentially pathogenic viruses. So you got to make sure that A, they're not going to hurt the doctors and the people that are working with them, and B, they're not going to hurt the people that you're injecting them into. So we've got to modify these viral vectors to make sure that they don't cause disease. Essentially what we do is we rip out any genes that allow the virus to replicate in the host cell. So it's really just a protein box filled with your piece of DNA in it, so that when it actually gets into the cell, it can still get into the cell because it still has the transfection machinery, it still has the capsid, but it doesn't have the ability to replicate, and it's the replication that causes disease symptoms. Hence, you can make them safe by completely inactivating them, in essence. So that's what we want to do there. And because of that, they're also not transmissible. So somebody that's infected with a viral vector can't get somebody else sick, can't pass it to somebody else because they're not producing active viruses. You want them to be or have low toxicity. A lot of viruses produce proteins that mess with the interior of the cell because cells try to fight back. And so there are viruses that can shut down the interior of, or that can shut down cell host protein production. I mean, viruses kill cells, right? We don't like killing cells when we're trying to help people. So we want to try and modify these viruses to make sure that they have as low toxicity to the host cells as possible. Stability, there are viruses that like to change a lot. For example, flu, we learned about it. It's got a segmented genome. Segmented genome means its genome is broken up into 9 different pieces. So in order for a flu virus to assemble, it's got to get all 9 pieces in there. And sometimes if you've got 2 different flu viruses that land in the same cell at the same time, they'll scramble up which segments they get, and then you get a brand new flu virus. That's why you have to get a new flu shot every single year. If I'm trying to build a viral vector that's got my gene of interest in it, I don't want to cross my fingers and pray that all the right pieces are going to assemble into the vector. That's not good. Right? So we want to make sure that we pick a virus and design a virus so that it has a low mutation rate and so that it doesn't scramble its genome. So we'll only use certain viruses, not other ones. And we'll talk about what specific viruses we use later on. The last one is cell type specificity. Occasionally, you want to try and deliver a gene to as many cells as possible. And we do use, there are circumstances where we will use viruses that will target a wide variety of cells. However, in most cases, it's a bad idea to deliver a gene to cells that aren't going to use it, that you don't want to use it, because then you just risk harm without actually doing any good. So we want to try and design viruses that can deliver the gene exactly to the certain cell types that we want to deliver it to, and not to any of the other ones. And viruses have this marvelous ability of tropism. They target certain cell types and not other ones. So we can hijack that ability to make sure that our target gets only where it's supposed to go and not anywhere else. Of course, these are the ideals. We don't always end up following them. Reasons for using viral vectors. So those other transfection mechanisms of getting genes into cells, we've got other versions. We can use transformation, where you put it on a plasmid and put it inside the cell. You can do electroporation, where you electrocute the cells and it makes little holes in the cell wall and gets it through. There's all kinds of things we can do. The problem is, what do you do on a multicellular organism like me? You can't electrocute me. I don't stand well to being electrocuted. It doesn't work all that well. So that works on individual cells, but whole organisms, it's very difficult to get into multiple cells. However, viruses are designed to do exactly that. They can infect multiple cells in an organism that's fully alive, and you don't have to electrocute them. It's wonderful. So we like using that ability for multicellular animals. Downside is it only works on animals. Plants don't have this, or we can't use viral vectors on plants. because plant viruses, which we don't get to talk about in this class because Dr. Lane doesn't like them, plant viruses are fascinating because they break a lot of the rules. They don't have spike proteins. Because if you think about it, structurally, other than having chloroplasts, what do plant cells have on the outside that an animal cell doesn't? A cell wall. It's this big, huge, thick armor plating on the outside. What protein receptors are you going to use? You have to get through miles of thick cell wall to get to a protein receptor to be internalized. It's not going to work. So how would a plant virus get inside the cell using a spike protein? It can't. So plant viruses have to use insect vectors, where the insect bites the outside of the plant cell and punctures the exterior and then drops off the virus on the inside. And then viruses don't have to get from cell to cell because all plant cells are connected. So once you infect one cell, it infects the whole tree, which is why plant viruses are so difficult to contain. Problem is, if we're using a bug to drop off the gene, that kind of defeats the point of using a virus to drop off the gene. So that's why viruses don't work on plant cells, or viral vectors don't work on plant cells. So we just use them on animals. And with bacteria, I'm sure you could do it with bacteriophages, but transformation with plasmids is a heck of a lot more efficient. It just doesn't work on animal cells. So, but that's one of the advantages that we like using viruses for. Virus tropism, as we talked about earlier, allows you to deliver genes to very specific cell types. And it's very, very good at getting inside cells. Viruses make up or survive by getting inside cells. And so we can take advantage of their efficiency because it will get the gene into a lot of cells in that person. There are challenges, however, that we have to overcome with using viral vectors. One, you have to make sure that the gene gets delivered to the proper cells in the proper individual at the proper time and in the proper amounts, because how do you prevent multiple viruses from infecting the same cell? And once it's in there, how do you make sure that the cell actually builds the protein you want it to build? You have to be able to control that as well and make sure it doesn't produce too much of it, because if you produce too much of the protein or too little of the protein, you'll either have harmful effects or no effect at all. So you have to control which promoters you're using. You have to think through how to get just the precise amount of the protein. Plus, you want to make sure that it sticks around. Most viral infections last about a week and then it's over and done. I want my cure for your disease to last longer than a week. So we have to think about these things, and there are some difficult hurdles we have to get over, and we'll talk about how you do that later. Immune response. It turns out the immune system doesn't like viruses. We have had problems in the past. So viral vectors have been used in clinical trials since the 90s, and we've had some problems. In 1999, this individual, his name was, holy smokes, I just spaced his name. I wrote it down. Jesse Gelsinger. I'm sorry. Jesse Gelsinger, in 1999, he participated in a clinical trial to try and cure his genetic disease. It was a minor genetic disease. I think he was 12 years old. And so they injected him with a viral vector, and 3 days later, he was dead. His immune system overreacted to it. He had a complete system-wide immune response, and it killed him. We've had all kinds of problems with that. So how do you make sure that the immune system doesn't flip out and kill the person that you're trying to help? We also have the problem that once you drop off the gene in the cell, certain cell types can actually methylate the gene you drop off and permanently shut it down. They'll recognize it as foreign DNA and prevent it from ever being transcribed ever again. So you're like, well, great, now what? So we have to worry about both an extracellular and an intracellular immune response, and we've got to try and find ways to get around that. We'll talk later, there are viral vectors that have the ability to actually glue their DNA into your chromosome, which is awesome because it makes a permanent change and hypothetically will cure you for the rest of your life. The problem is we can't always control where in your genome this thing gets stuck. So what if it slaps it smack in the middle of an oncogene and poof, I cure you of hemophilia and give you cancer in the same shot? Or give you a different genetic disease, right? So this is a problem, and we have had this happen before. They had a case where they were trying to cure some babies of what they call bubble boy syndrome. It's severe combined immune deficiency disease. Basically, these guys have to live in a bubble for their entire lives because their immune system doesn't exist. So if they get anything, they're dead. So they were trying to cure them, and it worked. And then a couple of years later, these kids got leukemia. and they were able to save all but one of them, I think, with chemotherapy. But we have to worry about that too. Commercial viability. I know this is the downside of medicine, but companies have to be able to make money making these things. The problem is most genetic diseases that are single gene genetic diseases that you can actually fix are rare. So you're going to spend millions of dollars trying to develop this medication to cure 6, 7, maybe 100 people. How are you going to make your money back? So it's difficult to get companies to invest in the technology. And of course, we'll talk about later, the production of viral vectors is extremely difficult in some cases. It's difficult to upscale it onto a broad production line scale. Okay, so now we get into the fun nitty-gritty stuff. These are the 3 major classes of virus that we use as viral vectors: adenoviruses, lentiviruses, and adeno-associated virus. And we're going to go through the specifics of each of these 3 in order here. I put them in this order because, as I understand it, these are the order that they were developed as viral vectors. So they started using adenoviruses first, and then they put lentiviruses, and adeno-associated viruses are relatively recent on the scene. Okay, so let's go into adenoviruses. Examples of adenoviruses are some of your common cold viruses. There are other viruses that can cause this, but a lot of adenoviruses cause common colds or pneumonias or things like that. They are naked icosahedral viruses, which we learned what that means. It means they have no envelope and they're shaped like an icosahedron. This is an electron micrograph over here in the corner of an adenovirus. It's kind of blurry, but you can see like a little triangle there. You can see one of its sides if you look really closely. It's got linear double-stranded DNA. Its genome size is about 35 kb, which is relatively big. And the insert capacity is about 8 to 35 kilobases. That's the size of the gene you can put in this thing as a vector, which is the biggest of them all. That's one of the advantages of using an adenovirus vector, is this thing's flipping huge. So you can cram, depending on the generation of adenovirus vector which you use, which we'll talk about, you can actually cram a decently sized gene into this thing, which is a great consideration because remember, you usually have to incorporate more than just the gene. You have to incorporate promoters, you have to incorporate enhancers, you have to incorporate a terminator. There's all these structural elements that you have to put around the gene that you have to send to so that the body knows how to turn the thing on once it gets there. Okay. So here we go. So this is what an adenovirus genome looks like. That was me. Thank you. This is what an adenovirus genome looks like. And it looks crazy and mayhemic because we haven't really gotten into the details. It's actually not that bad, and we're not going to go into a ton of detail. Basically, if a gene starts with E, that means it's an early gene. Usually viruses do gene production in 2 different stages. So an early gene usually has something to do with genome replication and protein production. Whereas a late gene, the ones that start with L, usually has something to do with production of the capsid, because you want to replicate your genome first and then produce your capsid around the genome. So that's why you've got an early stage and a late stage. So if you see something early, it's usually got something to do with DNA replication, which means these are the guys that we don't want in our viral vectors, because DNA replication leads to virus reproduction, and you don't want them to make babies in the cells. So the first generation vectors, they decided to come in here and erase the E1 gene right here. E1 is a critical gene necessary for the cells to or necessary for the virus to reproduce. It's kind of an activation factor that kicks off everything else. So when they eliminated that, that gave us a little gap to slap in a transgene, because you cut out a DNA. And of course, the problem with these vectors is it's a solid, rigid protein box. So you only have a certain amount of room in there. So you have to cut something out to put something in, if that makes sense. So the more you cut out, the bigger the transgene you can put in. So we cut out that one, and the advantage to these first generation vectors is they're really easy to produce. All you have to do is have a cell line that can have that E1 gene incorporated into it, so it produces the E1 protein, and then you produce these viral vectors in addition, and we'll show you how that works. The downside is it produces a heck of a lot of other virus proteins, and immune systems don't like virus proteins. So this is the viral vector that killed that kid. So we try to avoid that. But some first generation vectors are still used, but not as much because they have high immunogenicity. So instead, we want to pop to second generation ones. What we did here was we eliminated all of the E's. So now it's just the L's, which is good because these are just the viral capsid proteins. And that also means you can incorporate a bigger transgene. The downside here is they still got immunogenicity because these guys are still producing viral proteins. So they finally wised up. I'm like, how did it take you this long to figure out how to do this, guys? You just cut them all out. It's that easy. I don't even have a PhD in it. Sometimes people. I spent like a week trying to figure out why they did this, and I couldn't figure out why it took them that long. So this is the third generation vector. So you cut out virtually everything except the transgene. Notice we've got this little trident of Poseidon over here. That was a horrible circle. I like totally missed. Guys, it's been a rough day. That is the Greek letter psi, and that means the packaging signal. So you want to keep that in there because that's the signal that says put this inside a capsid. So if you don't have that, then your gene's not going to get packaged inside the virus and you wasted your time. So we want to keep that, but we deleted everything else. So that is how you get up to 35 kilobases of insert size because you replace their 35 kilobase genome with a 35 kilobase insert. So now we get to talk about how you produce one. All viruses have to be produced inside a cell, right? That's the whole point. They have to get inside the cell and be produced. So we have to have a cell to produce these things. And usually we use what's called a packaging cell line. And they're single cells. Usually we use like HeLa cells or kidney cells or some kind of propagated cell line. So this is my little cell. I believe the adenovirus vectors are made in kidney 298 cells. So here's my little kidney 298 cell line. So what you have to do to produce an adenovirus vector, and I have to try and remind myself on how many plasmids because it varies from one vector to the next. Okay. So on this one, what we've done is they developed these kidney 298 genes to have that E1 gene in there. So here's that little E1. So they embedded this, that looks like E7. It's E1, pretend that's E1. So this cell naturally produces this E1 protein that these viral vectors are missing. And then we can take a plasmid, which are those circular loops of DNA. Holy cow, I can't draw for the life of me. And then you slap all of the viral genes that you need onto this plasmid. This one's called a transfer construct. Wait. Nope, this one's a packaging construct. Sorry. Packaging. So that's got all the viral genes, and depending on which generation, like if you're doing a generation 1, you don't need a packaging construct because all you're missing is E1. And then the transgene produces the rest. For the later generations, you would need a packaging construct. And then you need a second plasmid, and this is the really important one. This one is your this one's the transfer plasmid. So this is the one that's got the transgene on it right here. So the transgene is just whatever gene you're trying to deliver. Transgene, this is remarkably hard to read on this thing. So you got your transgene and whatever, so this is where your little psi packaging signal will be. So the nifty thing about breaking it up like this, because you might just wonder, why not just slap the entire viral genome on the transfer plasmid and we'll just copy the whole thing and get it over with. Problem is you don't want the whole genome incorporated into the virus. That's the whole point. So by breaking this up and making sure that the transfer plasmid is the only one that has the incorporation signal, that means you've got all of the genes necessary to build a virus, but only the piece you want gets incorporated into the virus. Because this is how you build a virus that can't make babies. Because how do you make babies that can't make babies? Right? This is how you do it. You put all the components in there and then only have the components you want packaged into the virus itself so that it is replication deficient. Okay. Pros and cons of adenovirus vectors. The adenovirus does not integrate into your genome. That's why when you get the common cold, it only lasts about a week if you're lucky. If you're me and my wife, we get it for like a month. But it remains episomal, meaning that it just floats around. It's not incorporating into your chromosomes. It's just there. which is really good because you can't insert the gene into the middle of something important and give yourself cancer. So that's good. It's got high gene expression. These ones have the highest production capacity of all of them. Like, for some reason, something about adenoviruses really makes them kick out proteins. So once they get into your cells, you're going to produce a lot of the protein, which may be good or bad depending on what you're trying to do. It has the ability to infect non-dividing cells, which is fantastic, because what if I'm trying to change your neurons? Some viruses can only get into a cell that's actively dividing. And nerves divide like once every 20 years. So how do you muscle cells, if we're trying to cure someone of muscular dystrophy, you're going to have to get inside someone's muscle cells. They rarely divide. So you've got to have a viral vector that can get inside a cell that doesn't divide. And these guys can do it. So that's a huge, huge upside. They have the biggest packaging capacity, that 35 kilobases, and they have a high transduction efficiency and a broad natural tropism. So we don't have to modify them all that much to get them to go where we want them to go. Okay, next one, lentivirus vectors. Sorry, I am racing here. We're doing good on time? Okay, good. Lentivirus vectors. So have you guys heard of a retrovirus? This is lentiviruses are a subclass of retroviruses. So we do use retroviruses as transduction or as vectors. The downside to your natural retroviruses is most of them can't get inside the nucleus unless the cell is dividing. So most of them can only affect dividing cells. However, lentiviruses are a subclass of retroviruses that have the ability to get into the nucleus on an undividing cell. So that's why we prefer to use them in most cases. An example is HIV. And actually, HIV, like the virus that causes AIDS, is the most common lentivirus vector that we use because we understand it very, very well. They've done a heck of a lot of research trying to cure AIDS. And so we understand it very well, and it's very good at its job. That's why we haven't cured it yet. It's an enveloped virus. It's got linear single-stranded RNA, and it's reverse transcribed, which we'll learn about how that works later. It's pretty wild. It's got a much smaller genome size. Notice the adenovirus vectors could handle genes up to that were 8 to 35 kilobases. These guys can handle at max 8. So that's one downside, is we're starting to shrink in size as we go down because they're just physically smaller viruses. And that's an electron micrograph of HIV. Notice they've got this, it looks like a Mike and Ike. You're never going to look at that candy the same again. Tasty HIV. Okay, so here's the standard lentivirus genome. I pulled this one right out of the book, and it looks absolutely crazy. I'm not a big fan of this map. But once again, you got your incorporation signal over here. On this one, the most important genes that you need to produce a virus are the GAG, PUL, and ENV genes. This is the polymerase right here, the reverse polymerase that they need to do their reverse transcription. The GAG is the rest of the proteins that are needed for genome replication. And then the ENV proteins are for the outside of the virus. And then a couple of these other ones are important as well for gene transcription and whatnot. But on this one, we're not even going to go through all 3 stages. These guys got Ys, and they actually just cut straight to generation 3. And so we just cut out everything except your incorporation signal. But that does mean that you need, that when you're producing them, you have to have a couple of inserts. Now, HIV is a little worse than adenovirus, because adenovirus, if we accidentally build a functional working virus, you get the common cold and you get mad at your doctor. If we build an HIV that works, you get AIDS, and then you're really mad at your doctor, right? So we have to be very careful with how we build these things. So once again, we're going to have our packaging cell, because you have to have a cell to build things. And then we're actually going to have 3 plasmids that we throw in. Actually, there might be 4. Is there a 4th? Yeah, there are 4, which sounds kind of absurd. But I think it depends on which version you're working with, but we're going to have 2 of the plasmids that actually have the genes to build a retrovirus on it. Now, why would we need 2 and not just 1? Any thoughts? Because on the last one, we crammed all of the genes onto the same one. True, but remember, this one's genome was 8 kilobases, and the last one, we crammed enough genes out of a 35 kilobase genome to make 1 plasmid work. So why would we need 2?
Make it go from a capacity of 8 kb to 16 kb.
We're not packaging these 2 inside the vector. Good thought, though. The reason is you need a GAG gene, a pol gene, and an env gene in order to build a working functional virus. And there's such a thing as gene recombination, where you can accidentally, or you can stick a plasmid in here, and it can do homologous recombination and build a fully functional HIV genome that then gets packaged. We want to try and reduce that as much as possible. So if you have 1 plasmid, you just need 1 recombination event to build a fully functional genome. If you break the genome of the virus into 2 pieces, you have to have 3 separate incorporation events in order to assemble both the packaging signal, which is on the transfer plasmid, and then each of the pieces that are on both of the insert plasmids. And so by breaking up the genome into multiple pieces, you reduce the chances of building an actually functional HIV vector, because you have to have the genes from this plasmid up here recombine with the genes from this plasmid down here, recombine with the genes from the transfer construct down here in order to build a functional virus, and the odds of that happening are so extremely low that it's basically safe. So that's why on this one, you technically could do it with just 2 plasmids like we did with the adenoviruses, but it's safer to break it up into as many plasmids as you can. So that's how that one works. So once again, you only put the packaging signal on your transfer construct down here. Whoops, it thought I clicked. You only put it on your transfer construct down here so that only that gene gets packaged by the cell into the virus and spit out. The other thing that's nifty about these guys is we can alter their tropism. Remember how it was an enveloped virus? Well, that env gene, that means that rather than the spikes being built onto the capsid, they're embedded in the envelope, which means they're flexible. So we can actually alter where this HIV virus goes by swapping out its spike protein. For example, HIV viruses normally only attack your helper T cells, right? And normally we want to transform things other than helper T cells, because helper T cells are part of the immune system and they trigger an immune response and make everybody mad, right? But what if I want to try and use a lentivirus vector to target the liver to try and cure you from hepatitis? Well, there is a virus called hepatitis B virus. What if we rip its spike protein off, because it's designed to target liver cells, slap it on an HIV virus instead of its envelope protein, and ta-da, you have an HIV virus that can go to the liver. So because these guys don't have the spikes actually built onto the capsid, they're a lot easier to pseudotype, is what we call that, to swap out their spikes and make them attack whatever you want, which is really nice. And that's partially why you would put the rest of the genes up here, like the GAG and pol genes, you would put them on one plasmid, and then you would put the envelope plasmid on the other one, so that you can just buy the standard retrovirus package plasmid from somebody and then build the one spike plasmid that you need to save work, right? And then you build your transfer one, and then you got them ready to go. Advantages and disadvantages. The advantages is it infects non-dividing cells, which is good, just like the adenoviruses. They can do dividing and non-dividing. They have a broad tropism because we can swap out their spike proteins and make them attack whatever we want. And their genome inserts into your chromosome. Have you ever wondered why if you get the common cold, you're sick for a week, but if you get HIV, you're sick for life? The reason is because so it's an RNA genome, but it reverse transcribes it to DNA, and then it goes into the nucleus and embeds it in your chromosome. So the only way to kill the HIV is to kill you, because it's your chromosome that is building the HIV viruses. So that's why HIV is so difficult to cure. But that's good in our case, because it will embed it into your genome, and you're permanent. And now when that cell divides, it will replicate the gene and give it to the progeny virus. With those adenoviruses, it will get diluted every single time the cell divides, because you only have the one copy. It's not embedded into the chromosomes, and our cells only replicate chromosomes. So this has the ability to be a permanent change. I can cure you for life, which is really nice, in theory, assuming your immune system doesn't go and methylate the thing and shut it down, right? Which they've found ways to try and get around that a little bit. Downsides are your immune system hates HIV, so it will attack it. And it's a random insert location. We can't control where this thing is going to stick. So every time you use a lentivirus vector, you're crossing your fingers praying you're not going to give the guy cancer. which rarely happens given that like 98% of your genome is junk DNA, but there is a possibility. Okay, last one. Adeno-associated viruses. These are the brand new ones that they've recently started developing since the late 90s. And these guys are just like a jack of all trades. They're like the magic virus. They're pretty awesome. So one thing is, I mean, the example is AAV2, which nobody's ever heard of, right? But it's relative. Does anybody have dogs? Have you ever had to have it vaccinated for a disease called parvo? That's a relative of AAV. So it's really deadly in dogs. It does absolutely nothing in humans. So it has a so the AAV virus is the human version. It's a parvovirus. But for some reason, as far as we have been able to tell, AAV cannot get you sick. It gets into your cells, but it has no symptoms whatsoever. So most of us probably have AAV. In fact, I think I read something that said like 60% of individuals tested have resistance to AAV already. Because everybody has them, they just don't know it yet. So that's one real big perk, because if we mess one up and build and accidentally build a functional virus, it's okay, because it can't get you sick anyway, right? It's an envelope nucleocapsid. This picture over here, just to show you, in the bottom left corner, you'll see these massive viruses. Those are our adenoviruses from earlier. And the little tiny circles that are next to them are the adeno-associated viruses. So these guys are the smallest of the bunch, which means they have an insert capacity that is a mere 4.5 kilobases. That is their biggest downside, is most human genes are bigger than 4.5 kilobases. So, I mean, there are ways you can get around that. You can cut out the introns. You can make these genes shorter. You can cut out domains of the protein that aren't as necessary for the function of the protein. So they have ways of cutting genes down to make them fit, but that is a severe downside to these guys. Okay, here's their genome. It's that simple. Isn't that wonderful? After all the craziness earlier, we just need to stare at this piece of art for a little bit, just appreciate it, maybe start crying, pass tissues around. Okay, so you've got your, sorry guys, I'm in a weird mood today. So you've got your non-structural genes that are helpful for producing viruses, and then you got your capsomere proteins that build on the outside of the cell. And then they have the, notice they've got these inverted terminal repeats. That's what these are called, ITRs, on both ends. And what that is their DNA literally forms like a double hairpin loop on the end there. So as it shows, it comes around here, and this is paired up with that, like that, and then it pairs up to itself across the middle here. These inverted terminal repeats are nice because they serve as the packaging signal and the origin of replication and the promoters. So they're just kind of a win-win situation, which means those are the only parts you have to keep. So we've got this 4.8 kilobase genome, and we can keep all but like, or we can remove basically all of it and just keep those inverted terminal repeats. So that allows us to slap the maximum size transgene in there, which again isn't all that big, 4.5. The production strategy on these ones is, once again, you're going to have your cell line, but these ones, that was the wrong color. That's okay. These ones, however, are tricky, because the reason adeno-associated virus doesn't get you sick is because it's a novel virus. Most viruses can produce their own babies once they get inside your cell, right? These guys not only need a host cell, they need another virus. This virus can't produce its own babies unless the same cell is infected with an adenovirus or a herpes virus at the same time, because it has to borrow genes not only from the cell, it has to borrow them from the adenovirus or the herpes virus in order to make its own babies, which makes these guys really, really interesting to try and produce. Because not only do you have to just put in a packaging construct with the adeno-associated virus genes in there, you have to put in genes from the hepatitis virus or an adenovirus in order to try and build this thing, which means you're also going to produce adenoviruses at the same time. Then you have to sort out what's an adenovirus and what's an adeno-associated virus so that you only give them the adeno-associated virus, which makes these guys really fun to try and produce. So you're going to have a couple of different things to incorporate. You've got your packaging, or you've got your packaging construct right here that's got all of the genes for how to produce the virus. And then you've got your transfer construct down here. Dang it, it did it again. Fix on the block. Okay, there we go. Transfer. And it's got your transgene on it, along with the inverted terminal repeats. And so there's your transgene. And then these guys, for reasons that I have yet to figure out why, they actually take it a first generation adenoviral vector. So those first ones that were missing just the E1. So this is our kidney 298 cell over here that we were using to produce those. So they take a first generation adenoviral vector and they slap it into this cell too. So that way, hypothetically, you won't get adenoviruses out the back end, when in reality you actually will, because all the genes are there to produce an adenoviral vector. So, you're actually going to have to slap in an adenovirus as well. And when you put in all three of these, you're going to get both adenoviruses and adeno-associated viruses. The nice thing is adeno-associated viruses are really, really stable viruses. You can like cook these things and they won't fall apart. Adenoviruses aren't that stable. So essentially, you can heat these things up, have all the adenoviruses fall apart, and keep your adeno-associated viruses, and then you do gradient centrifugation to get just your adeno-associated viruses, hypothetically, and you purify them out. But it's a process and a half, and it's really, really difficult to produce enough adeno-associated viruses to make it commercially viable, which is one of the downsides to these guys, because the adenoviruses, you can produce mountains of them. Adeno-associated viruses, they're a lot slower, and so it's difficult to get commercial amounts, which is one of their downsides. So the advantage is they can't get you sick. They can infect non-dividing cells as well. They have very low immunogenicity. For some reason, the immune system does not freak out at these guys as much as they typically do to other viruses. And they don't usually have an intracellular shutdown response, which is also nice. So most genes that are delivered with an AAV vector stay there, which is good. And it remains episomal, kind of like the adenoviruses. I said little to no integration. The natural adeno-associated virus actually has the ability to embed itself in the genome. And it's awesome because it only embeds in one spot on chromosome 19 called the adeno-associated virus spot. Like that's its name. It's adeno-associated virus 1 location or whatever, which is wonderful. The downside is you need those non-structural genes to make it glue there, and we cut those out. So we kind of lost its major advantage by building the vector, but it's the sacrifice we have to make in order to not make it only carry 2 kilobases of gene. So it largely remains episomal, though it does retain some ability, like 1 in 1,000 can glue into your gene. But the downside is, since we got rid of those non-replication genes, it's kind of random where it goes. So we kind of lost that upside too. So the changes may be temporary, depending on if it embeds in your genome or not. But the nice thing is these things can last in your cells for months. So it's long-term effects, which is good. Downside is it takes like 60 shots to get one of these. So that's no fun. All in one day, apparently, too. You like sit there and there's in the leg. That's no fun. They're very difficult to produce in large quantities, and they have a very small packaging size, which makes them difficult to work with. So applications of viral vector technology. We've talked about gene therapy. You can deliver a transgene to the cell, and they actually have some, after the failures in the 90s, we backed off a little bit, but they're starting to have some clinical trials come out. In 2019, they had a clinical trial where they successfully cured a handful of individuals of hemophilia, and they were still fine 2 years later, and still perfectly healthy. And like, that's a genetic disease that you live with for your entire life, and they cured them permanently. So there are things like that. They've got a couple of adenoviral vector ones. A lipogene to Parvovec came out in 2012 to cure some random, really small disease. But that one actually is no longer on the shelves because it only got approved in the European Union, not in the United States, because we're stubborn. And it cost more than they could actually make back to produce. And so they actually lost money on it and had to withdraw it from shelves. But the rest of them are doing pretty good. There's actually a new one that I just learned about called Valoctogene roxaparvovec. Who comes up with these names? But it was designed to cure hemophilia A, and it just got approval in the European Union last month. So that's exciting. It's coming out, and hopefully they'll get it in the United States. CRISPR-Cas9 delivery. We can modify, for those of you, is everyone familiar with CRISPR-Cas9? We have to, okay, good. Seeing a lot of nodding heads. Okay, good. For CRISPR-Cas9 to work, you have to actually deliver it to the host cells. And for that to work, so we've got, you can use like an adenovirus vector. You can actually use a couple of the smaller ones too, to deliver the CRISPR-Cas9 constructs to the cells so that they can go and do gene editing or things like that inside the cells. Actually, if I remember correctly, that hemophilia one that they did in 2019 was a viral vector delivered CRISPR treatment. They can do vaccines. The notorious Johnson & Johnson COVID-19 vaccine is a viral vector vaccine. So Pfizer used an RNA vaccine where they wrapped it in a lipid bubble and had that targeted to dendritic cells. These guys, their strategy was to put the RNA for the COVID spike protein inside of a viral vector and have that deliver it, which is partially why they got more immunogenicity than the other guys, and it had such a bad reputation. But a lot of the COVID vaccines were done in Russia. They have the Sputnik 5 vaccine. That one is also a viral vector vaccine. So these are up and coming vaccines, and they're getting a lot better at making them less immunogenic. And they can use these for anti-cancer therapy. It turns out that there are some viruses that have the ability to, or like they shut down the interferon response inside the cells, and so then they can take over cells and kill them, right? But we can go in and modify these. So rather than putting a transgene into it, you just modify the virus so that you take out whatever gene allows it to defend itself against the cell's immune response. And turns out cancer cells don't have the interferon response. So if these cells get inside a normal, healthy person, they get inside your cells will wipe them out before they can make babies. If they get in a cancer cell, they can go crazy and kill the cancer cells, which means in the end, you only end up killing the cancer cells and nothing else. So they can use viral vectors as an anti-cancer therapy, which is an up and coming thing. The only downside is you have to try and target it to the cancer cells, which means it's kind of a case by case basis, which they're still trying to work around. But questions? Sorry, that was an awful lot of talking. Yes.
Okay, so you said like 60% of the population is resistant to AAV.
Yeah.
So would they need to test someone to see if they're resistant before they inject them with it to not provoke an immune response?
Hypothetically, the nice thing about these ones is they don't produce a super bad immune response. So even if so the worry about injecting you with one is A, yeah, maybe we could hurt you with an immune response, but B, your immune system might take them all out before they actually get in your cell and do any good. But thankfully, there are a couple of different serotypes of adeno-associated virus. So we just have to find out which one you're not affected with, or which one you've never seen before, and then infect you with that. But that is one of the downsides to adeno-associated viruses, since they're so proliferative. How do you find one that they've never seen before? Exactly. Great question. Other questions? Yeah.
So in the case where the genes were modified and that patients ended up developing leukemia, after going through chemo, would they still continue to develop leukemia because their genes weren't changed to like not have, I don't know, to not be blocked so that they don't produce leukemia?
Well, that's a great question. Hypothetically, all of the cells, so hypothetically, this thing wouldn't embed in the wrong place in every single cell that it targets, right? In some cases, it's going to embed in the right one. So because, I mean, all cancers are an alteration in your genome. So the only way to kill the cancer is to kill all the cancer cells, right? So essentially, the one or two cells that had it embedded in the wrong spot would start proliferating and you'd get leukemia or whatnot. So then the chemo would hypothetically kill off those cells and all of their progeny, but leave all of the healthy ones that had actually embedded in the correct spot. So hypothetically, if you survive the chemo, then you would still have the cure. Other questions? All right. Well, thanks for putting up with me today, guys. We'll let you go a bit early. Actually, we're right on time.